The novelty of electron microscope from others. How electron microscope is different from other microscopes, ordinary light microscope. In ordinary light microscope, the light is used, photons are used. But in case of electron microscope, the beam of electron is used instead of beam of light. Electron magnetic lens is used instead of ground glass lenses. In place of ordinary glass lenses, here the electron magnetic lens, which is a special type of lens, it is used in electron microscope. Cylindrical vacuum column is used. We will see later in the structure of electron microscope. This cylindrical vacuum column, electron should travel in vacuum to avoid collision with air molecule that cause scattering of the electron, distorting the image. So air should be absent when the electrons are traveling, because with the air molecules the electron will collide and they will scatter, and the a clear image of the material will not be obtained. So that is why cylindrical vacuum columns are used. Comparison of lens system of light and electron microscope. This is light microscope lens system, and here this is electron microscope lens system. Here is condenser lens of both, object lens of both, intermediate image, and ocular or projector lens. Viewer screen with final image. So the lens position is almost same, but the lens nature is different. Commonly used electron microscope in biology and in material chemistry, the same electron microscope are used. Scanning transmission electron microscope, which is also called TEM. Scanning electron microscope, the short form is SEM. Many for various life forms and microbes. Mainly for various life forms and microbes, for living materials and by microbe, as well as for inorganic material it can be studied. And there are other types of electron microscope, that is scanning, tunneling microscope, atomic force microscope, and in this the actual visualization of molecules and individual atom also in motion. We can see the atoms in motion, molecules in motion with the help of scanning, tunneling microscope and atomic force microscope. So first of all, we will study in detail the TEM, that is transmission electron microscopy. This is the image of transmission electron microscopy. Transmission electron micrograph of some well-formed gibbosite clay crystal. This is some type of crystals whose structure is here in transmission electron microscope. This is the image of transmission electron microscope. It is connected with a computer and the image will appear on the computer. The first TEM was built by Max Knoll and Ernst Ruska in 1931, with this group of developing the first TEM with resolving power of greater than that of light microscope in 1933 and the first commercial TEM in 1939. Transmission electron microscope definition. Transmission electron microscope is a microscopy technique where a beam of electron is transmitted through the ultra-thin specimen. The specimen is used which is in very thin form. So the electron will be transmitted from this thin specimen, interacting with the specimen as it passes through. It also interacts with the specimen. An image is formed from the interaction of the electron transmitted through the specimen. The image is magnified and focused onto an imaging device such as fluorescent screen on a layer of photographic film or to be detected by the sensor such as CCD camera. So there are different types of devices for showing the images and how image is formed, due to what type of interaction the image is formed. It is formed by the interaction of the electron which are transmitted, interaction of the electron which is with the specimen, those electrons which are transmitted from the specimen. And the image can be seen on different images devices such as fluorescent screen, photographic film or CCD camera. Application of transmission electron microscopy. Transmission electron microscopy are capable of imaging. Transmission means the electron are passed from the sample. They are not reflected or reflected back or scattered back. Some of the electrons may be reflected back and scattered back, but most of the electrons are transmitted from the specimen. Here, the application TAMs are capable of imaging at a significantly higher resolution than light microscope, owing to the small de Broglie wavelength of electrons. Because the electrons have very small wavelength, and due to which the microscope resolution will be very high. The wavelength of electron is very small as compared to the wavelength of photons. To examine fine detail of the structures in STEM, even as small as a single column of atom, which is tens or 1,000 times smaller than the smallest resolvable object in the light microscope, we can see very small particle in TEM, that is tens of 1,000 times smaller than the smallest resolving object in light microscope. Application in biological science like cancer research, virology, material science. We are studying inorganic chemistry course which is related to material science, as well as pollution, nanotechnology, semiconductor research, application in chemical and physical sciences like chemical identity, or to study chemical identity of the material, crystal orientation of the material, electronic structures of the material, and sample induced electron phase shift, and that is the changes which are taking place in the material, as well as regular absorption based imaging. Regular imaging which are based on absorption of electron. These are the application of TEMs. Here is another advanced type of TEM, transmission electron microscopy, that is called high resolution transmission electron microscopy, HRTEM. With the help of this, we can study the crystal structure. Crystal structure can also be investigated by high resolution transmission electron microscopy. HRTEM is also known as phase contrast microscopy. In a specimen of uniform thickness, the images are formed due to differences in phase of electron waves, which is caused by specimen interaction. Due to differences in phase of electron waves means when electron interact with the specimen material, the electron which are coming from the specimen, these electrons will have differences in their phases of waves, because they are passing from the different parts of the material, and due to differences in the phase of electron waves, which electron waves are coming from the specimen, from these differences the image of the specimen is formed. Image formation is given by complex modulus of the incoming electron beam, and the image is dependent on the number of electrons hitting the screen. Means the electron which come from the specimen, they will hit on the screen. For example, it is fluorescent screen. A more number of electrons hitting on the screen, a more clear image will be formed. And here is the mechanism, complex modulus mechanism for the image formation. It can be manipulated. The image can be manipulated to provide more information about the sample, as in complex phase retrieval techniques. So from the images we get the results, we manipulate the images, we get the results from the images. By taking multiple images of a single time sample at different angles. For example, this material we can rotate it at different angles, and we take the image at different angles. Typically in 1 degree increment, a set of images known as tilt series can be collected. Tilt means it is rotating slowly. This methodology was proposed in 1970 by Walter Hope, scientist, under absorption contrast condition. This set of images can be used to construct a three-dimensional representation of the sample, because when the sample is tilted at 1 degree angle, its all dimension can be visible in this tilt series, so we can construct a 3D representation of the sample.